Hi Nat5, so we are finishing off the Life on Earth unit with speciation. Okay, so what are we looking at in terms of the theory here? Now what we're saying is that we are looking at evolution as a gradual change in species over time. And as that changes, eventually you end up with a species that is so different from the original species that we say that's now a new species. And that's what speciation is, okay, the creation of a new species. Now, we are saying it's gradual and we are saying over time, but it's gradual in terms of each generation has a tiny change. Now, if we look at us, then you would say for humans, your generation time is years. But if you look at something like, say, a bacteria, the generation time might be 20 minutes. So we can see evolution in action in certain cases where you have what's called high-speed evolution driven by particular pressures. Okay, and that's really important, that you need a pressure. You need something that's going to actually create the selection which allows it to change. So all of this links in together. The concept of having mutations which allow changes to be there, variation, adaptations being the things that you might need to make to be better in a particular environment, and then the pressures that will drive that to cause natural selection, and then over time potentially speciation. Okay, just to remind you of something that you may remember from way back, second year or something like that, you get different types of rock, and one particular type of rock is formed by sediments. So that's tiny bits of sand, mud, things like that falling to the bottom of any river, water of any form, and then as more and more things come down off the land, it builds up, and then you get layers, and the layers get squeezed until they form rock. Okay, and you can see these layers in certain places, but what's really, really useful about these layers is that they can trap things inside them. The things that they trap for us that are useful are fossils. So fossils form when you get a living thing that doesn't fully decompose, and then over time, when they're inside the sediment, they get changed into fossils. And that tells us so much about what's happened, because if we can figure out where that fossil came from in terms of how long ago, then you can use that information to try and figure out where things actually were at the time and what they were like at the time. So that's referred to as the fossil record. So it's where it's found and what we can see. And you can use it to work out how organisms have changed, and even when things were around and when they became extinct. So here's just an image of a particular set of ones. These are all hominid skulls. So human is N in the bottom right hand corner, modern chimp is A, and we've got kind of what's happened in terms of changes, and we can see the hominid skulls moving through from that. So other evidence for change is actually the similarities that we can find. So we can look for appearance, so phenotypic similarities, and you can find similarities between different species. And you can also look for genotypic, so similarities. So you're looking at the genetic code and working out how similar particular species are to other species. And that's like a whole branch of science called phylogenetics, and really quite fascinating, and is part of the higher course. Okay, and we can actually see it happening. As I said when we were looking at earlier about the bacteria at 20 minutes per cycle, as it were, we can actually show evolution in bacteria just by pushing them using things like antibiotics. So to remind about this, for evolution to take place, you need to push, you need something to select for a particular change, and that's the whole natural selection, survival of the fittest. And the selective pressure is called a driver. Okay, now this is from effectively a recap from the first key area. First big driver, competition. So that's quite obvious. If you're better at getting a resource, you're more likely to survive and pass on. Okay, so any of the things that we had, and you could go on for ages. Disease is a really big one. Okay, so if you're more resistant to disease, then you're more likely to survive, you're more likely to pass on that immunity. Okay, so at the moment, obviously, we are very aware of this. Predation is a big driver. So anything which is better at escaping from the predators is more likely to pass on what allowed them to survive. So it may be a physical change, that they have a particular allele which allows them to move faster. But it might be something about sneakier, it might be better camouflaged, it might be a behavioural one, which means that they're more likely to stay closer to, I don't know, a nest or a burrow. I mean, there's so many different ways that you could be better, but the most important thing is, if you survive, that it's something you pass on. And back to that high-speed evolution, the idea of the superbugs. So MRSA, particularly nasty superbug that we have in our hospitals now and in other places, is really quite worrying because it is completely resistant to some of the main antibiotics that we use. And if we want to prevent the spread of this, this is a really, really big question. I mean, it's massive. How do we stop this being an issue? Because we are producing the pressure to make this evolve. If you have, say, 1 million bacteria, and obviously there will be variation within that bacteria, and maybe one of them is resistant to a particular antibiotic, you then use that antibiotic, you wipe out every single one of the other ones except for that one. And actually what you've done is you've wiped out all its competition. So it will happily start to divide, spread, and it will pass on its resistance. And if you do that with several antibiotics, then you can end up with a multi-resistant one. On top of that, there's even more worrying stuff about the fact that bacteria can pass resistances on back and forward. So, two types of evolution. We have divergent evolution, where things spread out from a common ancestor, and that is the one you need to know. Just to tell you, there is another kind called convergent evolution, where you end up with similar structures from a different starting point. Some of them are quite cool, like our eye structure is very similar to an octopus eye, but we didn't come from the same common ancestor. Well, not recently. So adaptive radiation, the one that you need to know, divergent, is where you start from a single common ancestor and then adapt to different food sources, in this case for Dartmouth finches. The Pentadactyl limb is a really nice one as well. So there's a basic 5-fingered limb on a huge number of animals on the planet, okay? And although they all look very different, if you kind of take them apart and look at the anatomy, we all have the same stuff inside. It's just adapted quite drastically for different niches. Okay, so how do we go about the actual process of speciation? So to start with, let me re-get in our heads the definition for species. It's a group of individuals that can interbreed to produce fertile offspring. No option, that's your definition. So speciation is just the development of a new species. So let's run through the system. Need a population. Okay, and here's my population. Can't remember what I call them. I think I went with mugwumps last time. Okay, so we have here just a bunch of little dudes. Okay, it's a whole population. They're all mixed. That's it. So first stage in speciation is isolation. And by that what we mean is that you need to take a population and then you need to divide it with a barrier. So you need to be able to give 3 potential examples of types of barrier and then what they could be. So our 2 types are potentially, 3 types, geographical, ecological and behavioural. So geographical is the obvious. You need a mountain range in the way or the sea, something big, okay, physically big. Ecological means that you have one of your abiotic factors that's creating a barrier. So something like temperature, humidity, pH are given here. So you could have, say, a lake where you have a pH barrier in the middle of it. And that pH barrier means that one group from one end of the lake cannot get to the other group at the other end of the lake. Something like fish, okay? It could be behavioural, and that's probably the most complicated one. So this has to be something where, although they're living in the same place, there are 2 groups that then become part of that same space that don't breed with each other. So for example, it might be ones that are active at different times of day. The other example we've got up here is that breeding seasons go out of sync. So you might have a group that are breeding at one time of year and another one that's breeding at a different time of year. And if they are breeding at different times of year, then obviously they are distinct groups, they're not going to interbreed to produce fertile offspring because they're not breeding together. Okay, but at the moment they're still the same species, because if you manage to get them together again, they would produce fertile offspring. Okay, so here are my groups split up in isolation, and what's really important is that we have no gene flow. So there's no individuals that are actually communicating alleles from one side to the other. Okay, so now what you need is time, and in that time what we need is for variation to build up. Okay, so what we have now are two populations which are distinct in the differences they have. Okay, now if we took away that barrier right now, there hasn't been enough time for the species to change. So this species is still one species, but we have subpopulations of each one, and each subpopulation now has a very distinct gene pool. There will be a lot of genes that are the same, that have not changed, but they have ones that are different now. And then we have natural selection. So for natural selection, what we need is for those two subpopulations to have different selective pressures. So in this little example, we've gone for, on this side, we've got tall grass, and our tall dudes can survive well because they can see. Okay, so that becomes more prominent on that side. On the other side, it's colder, so it is the little hairy ones that are much more better insulations, and so therefore they survive. So now you're starting to get a difference in what's happening with each of these groups. And if you leave this for a really long time, keep on pressuring, keep on using natural selection, eventually, and it is a really long time because it has to be a lot of generation, speciation takes place. And now even if I bring together these two populations, they might be close enough together that they could actually interbreed for one generation, but that generation is infertile. That tells you they must be different species. And that's it.